Good morning one and all. In this video, we will see about what is recombinant DNA technology. What is a recombinant DNA? A hybrid DNA, a chimeric DNA of a foreign DNA with a vector DNA. What is rDNA technology? Production of recombinant DNA by inserting into a host cell for maintenance or gene manipulation. What are the uses of recombinant DNA technology? Gene cloning, gene manipulation. Why is recombinant DNA constructed? To protect the foreign DNA from the nucleus of recipient cells, to make the foreign DNA replicate along with the vector DNA, to have genetic markers helping us for the selection of recombinants, to enhance the expression of the desired foreign gene in recipient cells. The first step in the construction of the recombinant DNA is preparation of the desired gene. The desired gene can be taken from the genomic DNA. So we have to isolate the genomic DNA. For this, the bacterial cells are washed with NaCl and water, then treated with SDS so that the cell membrane is disrupted, and the lysate is treated with proteinase K so that the cellular proteins like histone which are bound to DNA is digested. After centrifugation, the supernatant is treated with phenol chloroform mixture to precipitate the protein part. Again centrifuge and the supernatant is treated with ribonuclease so that the RNA will be digested, and again centrifuge and treated with acetate ethanol solution. When stirred well with a glass rod, DNA forms a white precipitate. The precipitate is washed with tris EDTA buffer and then with ethanol, sodium acetate, magnesium chloride solution and suspended in TE buffer. The second method of obtaining the desired gene is from mRNA. Insulin mRNA is isolated from the islets of Langerhans of the pancreas. Fractionation of the mRNA is done using gel electrophoresis. The mRNA is treated with reverse transcriptase and dNTPs, and the complementary DNA is synthesized. After this, treat with alkali to remove the mRNA, and then cDNA is treated with DNA polymerase and primer so that the duplex DNA is synthesized, and this DNA is used for the construction of recombinant DNA. The third method of preparing the desired gene is from the free nucleotides. Small oligonucleotides can be synthesized from free nucleotides using a gene machine. After obtaining the desired gene, the second step is the preparation of the vector DNA. Vector DNA is a DNA that carries a desired DNA into the host cell. We have different types of vector DNA as plasmid, gosmid, plasmid, phagemid, bacteriophage and artificial chromosomes. To isolate the plasmid DNA from E. coli, cells are treated with lysozyme and EDTA and the cell wall is ruptured. Spheroplasts are treated with sodium lauryl sarsenate. Cell wall debris and the proteins are centrifuged and removed off. Then cesium chloride density gradient centrifugation is performed and the plasmid is obtained in the lowest layer, and it is used to construct the recombinant DNA. The third step in the construction of the recombinant DNA is insertion of the desired gene into the vector DNA. The vector DNA and the foreign DNA are cut with restriction endonuclease. They are mixed and the nick is sealed by the enzyme DNA ligase. The fourth step is introduction of the recombinant DNA into the host cells. There are several methods available, and the natural methods include transformation, transduction, transfection, agrobacterium mediated, retroviral transduction, conjugation and transposition. Artificial methods include physical methods like microinjection, macroinjection, biolistics, protoplast fusion, chemical methods like calcium phosphate mediated, polyethylene glycol mediated, DEAE dextran and using liposomes, electrical method like electroporation and electrofusion, Novel methods using laser rays and sonication. Transformation is the uptake of DNA fragments or plasmids by a bacterial cell, and this takes place in the presence of calcium 2+ ions at 4 degrees C, and the temperature is raised to 42 degrees C by giving a heat shock. The ability of bacterial cell to uptake a foreign DNA is called competence. Recombinant DNA of size less than 15 kilobase pairs could be transferred using transformation. The next method is transduction. Introduction of a foreign DNA into a bacterial cell through a virus or a bacteriophage is known as transduction. Recombinant DNA of size 38 to 50 kilobase pairs can be transferred. Cosmids are plasmids with cos site obtained from lambda-phages. Cos site help in the packaging of the DNA into a lambda-phage vector. Lambda-phages have non-essential regions called the stuffer region, which could be replaced by the foreign DNA.